Hello, I welcome you to this another lesson where we look at the HIV structure, HIV cycle, and types of ARVs that we have. Okay, so my main topic of discussion is about the HIV cycle, and students have helped me come up with some acronym that would help actually remember all the 7 stages of HIV life cycle. Okay, so I want to put some meat to that. And then I've always told students that you cannot know the HIV life cycle if you don't know the HIV structure. So it's important you know how to draw roughly the HIV structure. All right, so to talk about the HIV structure, I would say that the HIV structure has got something like a hand, which we call which is part of the envelope, and we call it the GP 120. Of course, you are reviewing the diagram behind there. You are seeing those things. So we have the HIV. So we have something that is more like the arm, and then we have the hand. It is the GP 120, and also there is the GP 41. Okay, so those are part of the viral envelope. And then we have also the viral envelope, which is mostly made of fats. And then we have the GAG 17, that is just around the viral envelope there. And then we have the capsid, which is made of the GAG 24. And then inside the virus, we have the viral RNA because the HIV virus is RNA in nature. And in my drawing of the diagram, I try to show it more like there's a face inside there, there's a nose and the like. So the dots there are symbolizing the 3 enzymes that are found in HIV. And those 3 enzymes have put up an acronym there, which is TRIP. So that is for reverse transcriptase. It it's more like a backwards way of remembering this reverse transcriptase there. Then we have Integris and then P for protease enzyme. So these enzymes play a very cardinal role in the multiplication of HIV. And then the S looking like the strand there, that is the viral. That's a viral RNA. Okay. So to understand this topic also, you also need to understand from your biochemistry the formation of DNA, the nucleotides, those things that are needed to build up for the DNA and the RNA, which is also required, the messenger RNA, the T, whatever. So those things that are supposed to be carried to the nucleus so that you have a new DNA that is formed. So you have to revise those things, then you come back to this topic, then you will understand it very, very, very well. All right, so now let's go back to HIV cycle. So HIV cycle, we can remember it by using the acronym AFTI-RAB. So AFTI-RAB, that is summarizes all the 7 steps, and I've broken them down there to expand them so that you can understand what those letters stand for. Underneath there, I've tried to put the trip for the enzymes, which I mentioned, reverse transcriptase, Integris, protease. And then I've also mentioned some core receptors that are found on the host cell, that is the CCR5 and the CXCR4. Those are very important in helping further binding of the virus to the host cell so that the virus can find haven within the host. Okay, so A is attachment. So when you're talking about attachment within the life cycle, what is involved is that the GP 120, GP 41, they play a role. So they play a role in that they come in contact with cells that have got CD4 receptors. Okay, cells that have CD4 receptors have also CD4 core receptors, which we are calling the CCR5 and the CXCR4. So those are HIV core receptors. So that is what is necessarily happening within the attachment phase. Okay, so you have to mention the roles that these things are playing in order for me as the person who's like mentoring you or that is now needs feedback to understand that you got something from this. So GP 120 which is going to bind to CD4 receptors. When it binds to CD4 receptors, the CD4 core receptors, the CCR5 and the CXCR4, are going to help to for further binding and confirmation so that the virus now can find a safe haven. All right. Good. So then F is fusion. What is happening in fusion is that all the contents of the virus, all the contents of the virus are now emptied into the host cell. That is what's happening in fusion. The content of the virus enters the host cell. And what are these contents that you're talking about? The viral RNA and the three enzymes, the TRIP. Okay, so TRIP is just an acronym. Okay, then transcription. Transcription, that is the third stage. So transcription, here is an enzyme that plays a role. That is the reverse transcriptase. So the reverse transcriptase affects the transcriptions of the proteins that are needed for new DNA to be formed, and it will do a reverse process to make proteins that are needed for the formation of the virus. So we have the reverse transcriptase. The transcripts here, we're talking of proteins like the nucleotides, nucleosides that are needed for buildup of the new DNA. All right, then we have integration. So integration, we have this new viral protein that needs to be integrated to the host DNA. So the enzyme Integris is going to ensure that this new protein can be integrated, okay, or can be combined to this to the DNA of this host cell of the person who has been infected. Then we have duplication. So duplication is simply a process where the new viral proteins that have been formed are cut into smaller pieces that are needed for the formation of a new virus. So duplication here, we have the protease enzymes that is going to cut the new proteins that are now being formed, facilitated by the nucleus there, that are now being formed. They are going to be cut into small pieces that are required for the new virus to be formed. So when these proteins have been cut, they assemble. So that takes us to the next stage, assembly. They assemble at the bottom of the cell with all the needed pairs that should be there: the viral RNA, the three proteins, and all the other essential proteins that the virus may need, because the virus is protein by nature. So they will assemble together in readiness to form a new virus. Then we have budding. Budding, here we are looking at the process by which the virus now, as it comes out of this host cell, buds out. It buds out, makes more like an inflammation as it comes out of this cell, creating a lot of holes within this cell, making it weak. Hence, this will now lead to the reduction of CD4 cells, because viruses target CD4 cells. All right, so that is about the life cycle of HIV. All right, so now we have to look at the types of ARVs that we have. Okay, so the list is not exhaustive. There are more and more new antiviruses that are being formed. So ARVs for antiretroviral drugs that we are really talking about here. So we have fusion inhibitors. So fusion inhibitors will prevent the DP120 from fusing with the CD4 receptors, hence preventing entry of the virus into the host cell. So that is the mechanism of action. All right, then we have the nucleoside reverse transcriptase inhibitors. So now, when you look at all these drugs here that I've given you, they have an I at the end there. So the drugs that have been formed, they target their target is to inhibit a certain action, and the action is of the enzymes. Okay, the enzymes that play a role in the multiplication of the virus. So we have the reverse transcriptase there at the middle in the NRTI. But which building blocks are we trying to block? The nucleotide. So, so we have the nucleotide reverse transcriptase inhibitor. So it inhibits the formation of new proteins that can be used by the virus to build itself. Okay, then we have the NRTI. This one is different from these other drugs because it is not blocking any enzymatic action. The work of NRTIs is the info is non-nucleoside reverse transcriptase inhibitor. So this one is a pseudo or it's a force building block. It's a force building block in the formation of the virus. It's a force building block because when a person takes these antiretroviral drugs, they'll be recognized as a building block for the virus, but they are there to weaken the structure of the new virus that are being formed. Hence, you are not going to have a new virus being formed. All right, then we have the Integris inhibitor. So this one is going to inhibit the action of the Integris enzyme, which combines the new viral proteins to the DNA of the host. So the enzyme that does that will be inhibited. That is the Integris inhibitor. All right, then we have the protease inhibitor. So the work is to inhibit the protease from cutting those proteins that are needed for the virus. So we have that protease inhibitor. Please, I do not want to add the names of the drugs there and the likes because I really wanted my lesson to be short. So I encourage you to look at the drugs that are found in all these groups. I encourage you to read more because what I've just given you is a little bit of a brief of really what happens in the HIV life cycle. Review the HIV structure, review the HIV life cycle, and see how these roles come into play. So I've just given you the main points actually that happen here. Types of ARVs, and I can also quickly mention that in the combination of drugs, of antiretroviral drugs, where you have got two NN, there you have to take one drug. Okay, and then where we have one N, mostly we take two drugs. Where we have one N, you take two drugs. Where you have two N, you have to take one drug. So under NNRTIs, I just talk of EN, like a nurse with a certificate, enrolled nurse. So we have a fabulence and the verabine. And they all have the ver from the virus in the middle, the efavirenz and neverapine. Okay, so we have also the NRTIs and the likes. But otherwise, in my lessons for pharmacology, I'll try to briefly help you on how to remember the drugs and even ARVs will be covered. I thank you most sincerely for listening to this lesson. Please, if this lesson has been beneficial, please share and subscribe so that each time we produce another video, you are able to watch these videos. I thank you and goodbye.